Good afternoon, everyone. My name's Greg Fairn, and I'm a scientist here in the Keenan Research Center of the Shing Knowledge Institute as part of the St. Michael's Hospital. And today I'm going to talk to you a bit about some of the work that I've been involved in recently, but in a much more general term. So hopefully everyone can get a real sense for some of the problems that we're studying and investigating, but not with going into so many of the complex details. And so I'm trained mainly as a cell biologist and biochemist, but my work involves studying immune function, and so it takes on a lot of different angles: microbiology, immunology, cell biology, and so it really keeps me flexible in the type of studies that I do and techniques that are used. And so illustrated or highlighted here on the screen is an image of a white blood cell encountering bacteria. And you can see that the white blood cell is very dynamic, reaching out and touching some of the bacteria. It's going to end up taking them in and hopefully degrading them. Otherwise, the bacteria could end up causing disease in the host. That's sort of one of the major lines of investigation that I'm involved with. But the white blood cells in the body play a lot of additional roles beyond just immunity, including tissue homeostasis, cleaning up debris following cell death, and those sorts of events. And so they're very important cells in the body. And so I work mainly with macrophage, which is a particular type of white blood cell, and I'll be referring to them as phagocytic cells. And so they bridge both innate immunity as well as acquired immunity. And their role is really, in that sense, is twofold. They will phagocytose targets through receptor-mediated interactions. They will take up these particles and degrade them in phagosomes, and then they will end up presenting small molecules to other cells of the immune system. And so one type of cell would be a T helper T cell, and this can lead to adaptive immunity, so that then B cells can be activated and will start generating antibodies against whatever the macrophage has just encountered. So the process that I study mainly is referred to as phagocytosis, and really it's the process of cell devouring. And so you can think of it as cell eating. And so it was described in the 1880s by Michnikov. And so a very interesting guy. His life was certainly a bit of a roller coaster. He was married twice, but in both cases, his wives died very early in one case from tuberculosis and the other from typhoid fever. In fact, he had sort of become so distressed by the second death that he actually infected himself with the same typhoid fever. However, he actually survived and moved on. And after all of this happened, he was a professor this whole time, but he began studying sort of phagocytes, but not the white blood cells of the human body. He started out with starfish larvae, and these are translucent structures. And what he would see is that there were certain cells that just seemed to have no real purpose, but were always moving around within the larvae. And he thought that maybe these were some sort of sentinel cells that was just going around making sure everything was okay. And so what he ended up doing was taking thorns from tangerine trees, which are very small, and basically poking them into the starfish. And then the next day, he'd find that all these formerly motile cells were now surrounding the thorns, just trying to neutralize them. He then followed this work up by using an even smaller particle, spores from a fungus, and using plankton. And now this time you could see that actually the plankton cells were able to phagocytose the spores, and that's where the idea of phagocytosis sort of came about. And in 1908, he shared the Nobel Prize with Paul Ehrlich, who had previously described adaptive immunity. So in the same year, both the major component of innate immunity as well as adaptive immunity shared the Nobel Prize. So a few people in the room might know who this guy is. He's a famous baseball player and coach, and his name is Yogi Berra. And he's an Italian-American, son of immigrants, and his use of the English language, we could say, is very colorful. And one of his more famous sayings is that you can observe a lot by just watching. And really, that's what Michnikov was doing. He was combining two things and just kind of watching them. And it's been it's really what we still do a lot of in science and cell biology today. And so this here is another phagocytic cell. This is a neutrophil, sort of in action. And what you're going to find is that it's going to chase around this bar-shaped or dumbbell-shaped bacterium and finally going to engulf it. So I'll just play this here now. So you can see the neutrophil chasing around. At one point, it decides to chase after one bacterium and let the other one go. And you can see how dynamic and responsive these cells are. It's able to turn on a dime. And then finally, it actually catches up to the bacterium and is able to engulf it. And so these types of observations ask a lot of questions about how cells can move, change directions, phagocytosis, and so on. But you guys might remember from my title, I said something about how to eat something bigger than your head. And it's obvious that bacteria is not very big. So that's, it's impressive, but it's not spectacular, we'll say. So here is a movie that a friend of mine had produced. And so now these are 8-micron latex beads covered in antibody. And the macrophage here is only about 12 microns itself. So this bead is actually quite similar in size. And it's probably on the order of 4 to 5 times bigger than a normal bacterium. And so the macrophage is kind of just scoping the environment, runs into this bead, and quickly takes it up. And then another bead falls into place, and the cell again engages the antibody and is able to phagocytose the particle. And still not full yet, it's able to take up a third particle. So now the volume within the cell is much greater than it was before. And the other interesting sort of fact is the cell's not actually getting smaller. The plasma membrane isn't getting smaller as it eats these particles. So there's really a lot of membrane being delivered to the sites of phagocytosis because these particles are still surrounded within a vacuole or a lysosome. So I think just from some of these very simple movies, there's some pretty obvious questions that sort of arise. And the first being, how do cells physically eat? Where does the extra membrane come from? If the cell membrane, the plasma membrane isn't actually getting smaller, where's all this extra membrane coming from? What happens to the internalized particles? And then we've already seen opsonized latex bead coated with antibody. We've seen a bacteria. But how many sort of phagosomes can be produced, and are they all the same? So just a quick reminder for everyone that mammalian cells or animal cells are made up of different organelles. And all the organelles are membrane bound, whether it's the nucleus where the DNA is stored, or the lysosomes, which are sort of the cell stomach. They're surrounded by bilayers, the same that the plasma membrane is made up of in. And for this talk, the two main membranes that I'll be talking about is the cell membrane or the plasma membrane and the lysosome. So the plasma membrane is really how any cell sort of interfaces with its environment. It's how it senses stimuli and has to respond to changes in the environment. And so the membrane is made up of this lipid bilayer, and within it are different proteins, these integral membrane proteins, And then there are also peripheral membrane proteins. Just below the plasma membrane is this filamentous cytoskeleton that's made up of actin. And there's interplay between the cytoskeleton and the plasma membrane in that the cytoskeleton influences the activity of some of the proteins there, as well some of the proteins and lipids that make up the membrane influence the polymerization and depolymerization of the actin. And so actin itself is almost like a chain. And so it's made up of small individual subunits that assemble together sort of head to tail into filaments, and the filaments can interact with each other. And so you end up with a chain or a rope-like assembly. And this is important for what's going to come up in a bit, but the filaments are both assembled or sort of polymerized and depolymerized sort of constantly and at the same time. And so to look at molecules like actin and other proteins in the cell, we make use of green fluorescent protein, or GFP. And so this is useful because using fluorescent microscopy, we can excite this fluorophore with a blue laser, and we get off a green light. And so this makes it easy for imaging. And since the discovery of green fluorescent protein, labs have spent a lot of time making different mutants to change the different colors that are available. So if this is the green fluorescent protein here, people have developed other ones, and this particular one is referred to as M-cherry, which you basically get off a red light. And so here's a macrophage that's expressing an M-cherry tagged actin molecule, and what you'll see is that where the intensity is higher here, there's actually more of the actin protein. And so again, this is a phagocyte eating large particles. And you can see that as the eating goes on, that there's a lot of actin built up at the sort of pseudopods that are engulfing the structure. But then as soon as it's internalized, all the actin goes away. And so I'm not sure how well that projects, but this is a slightly different representation of the same type of data. Here, though, there's this sort of intensity scale. And so black means that there's no fluorescent signal. White is that there's the sort of highest levels of signal. So I'll just show, these are again macrophage expressing this M-cherry actin construct. And what you'll see is that whenever the cell eats a particle, there's this sort of flash of yellow and white. And that's because there's a real, I'll show that one more time. There's very localized actin polymerization helping to internalize the structures. And then once they're inside, the actin disappears. And so a molecule that I've studied a lot is actually a lipid found in the plasma membrane. It's called phosphatidylinositol 4,5-bisphosphate. Just giving you guys a little bit of the level of detail here in this slide and the next. It gets very specific and a little gritty if you're not sort of immersed in the field as to all these players and what's actually going on. But this lipid is a key regulator of actin polymerization. It both is a positive regulator of molecules that support actin assembly, as well as it inhibits molecules that disassemble actin. And so this way, the levels of PIP2 are very important for the actin assembly and disassembly. But it's actually far more complex. This is just some of the signaling pathways involved in actin polymerization and the internalization of antibody opsonized particles. And so ultimately, actin polymerization and actin dynamics is a key driver of this internalization process. So one of the other key components of the eating or the phagocytosis is where is this extra membrane coming from? And here's a movie that I made a few years ago now. There's a plasma membrane marker in green. There's sort of a generic membrane marker in orange. And so you can see where the two are, the color is yellow, but on membranes that are not the plasma membrane, it's just this sort of orange color. And this cell is going to eat 3 or 4 particles, all sort of in this region. And what you'll notice is that they stop being green very quickly and become just orange. So let's start that. I'll play that one more time, but you can see the plasma membrane sort of probing the environment with these sort of tentacle-like structures, but as soon as the particle is pulled inside, they're no longer made up of the plasma membrane. So as soon as it's inside, it's just orange. And so really, at the sites of phagocytosis, there's a lot of membrane being delivered. And the two major reservoirs of membrane, if we want to think about them that way in the cell, is the endoplasmic reticulum and endosomes and lysosomes. And so the two are illustrated here. The ER is surrounding the nucleus here, but also spreads throughout the cell. Whereas lysosomes are much smaller structures and are not interconnected like the ER, but still quite abundant themselves. And for the sake of time, I'm just going to say that a lot of work's been done, and there has been some dispute in the field, but it seems like the major source of the membrane is from the delivery of endosomes and lysosomes to the site of phagocytosis. The ER may play a minor role or may play an important regulatory role, but as far as the delivery of bulk membrane to surround that particle that's being internalized, it seems to be endosomes and lysosomes. So here's just a summary sort of little schematic of what's going on during phagocytosis. So if this is the plasma membrane of the cell, the white being the bilayer, the red being the actin underneath, an antibody opsonized particle comes in contact with the cell. It stimulates the polymerization of actin. The actin polymerization grows and starts surrounding the particle. At the same time, endosomes and lysosomes are delivered to this site, contributing to this membrane that's engulfing the particle. And then towards the end of the process, the pseudopods that are extending around fuse together to reseal the plasma membrane. The actin at the base of the cup has been depolymerized, and now the particle is pulled inside. But it remains surrounded by membrane itself. And so that basically takes us to this sort of stage where we have an early phagosome or a nascent phagosome, but what happens at this point? So within the cell, lysosomes really represent the cell stomach. They have low pH. They also contain a lot of proteases and lipases that can degrade the lipid and degrade if it's a bacteria or an apoptotic body or whatever the case might be. Cells of the immune system, the neutrophils and macrophage, the white blood cells, are also special in that they produce additional molecules to help kill invading pathogens. So one of the molecules is referred to as superoxide. It's a highly reactive oxygen species, but it has a very short half-life in the phagosome. It's converted either through a spontaneous dismutation or by an enzyme to hydrogen peroxide, which is common to everyone. If people have scraped themselves or cuts, this is the same hydrogen peroxide that you put on those sort of cuts and scrapes. The hydrogen peroxide can be used as a substrate to actually produce bleach or hypochlorous acid, which is, again, the same bleach used for making your white shirts really white in the laundry. And it's a very powerful antimicrobial substance. And so the combination of low pH, these reactive oxygen species, enzymes help macrophage and neutrophils degrade invading bacteria and pathogens. And so that's kind of summarized here. There's different enzymes involved. One involves pumping in protons, which is what's going to make the vacuole acidic. There's also this NADPH oxidase complex that produces the superoxide. There are other antimicrobial pathways that I'm not going to talk about today, involving reactive nitrogen and so on. But an interesting question comes up, well, if the white blood cells are so good at killing all these bacteria and different pathogens, why do people actually get sick, and why do we need antibiotics? And so through the power of evolution, a lot of the bacteria have evolved ways of either inactivating the superoxide or preventing the phagosome from becoming acidic, or producing a catalase enzyme that converts the hydrogen peroxide back into water. So pathogens or bacteria that are good at causing disease are also good at preventing the phagosomes from killing them. And so just highlighted here are 4 different bacteria that cause disease, and they actually use different ways to avoid killing. And so in the case of something like Mycobacterium tuberculosis, they actually block the acidification of the phagosome. So it stays more towards a neutral pH, and they're able to survive in this compartment. Legionella actually changes the way the phagosome matures, and so instead of fusing with lysosomes and acquiring all these enzymes and becoming acidic, it actually fuses with ER-derived vesicles. And so again, it stays at more of a neutral pH, it doesn't get all the proteases, and the Legionella bacteria is happy in that environment. Listeria actually takes a different strategy. It actually pokes holes in the phagosome and escapes and gains access to the cytosol, where now there's access to all kinds of nutrients, and again, none of the harsh molecules that normally kill them in the phagosome. And finally, Coxiella takes sort of a real different strategy in that it's fine growing at the low pH and just adapts. And so it's the sort of thing, it's like, you want to put me in a really harsh environment, they're sort of like, we don't care, we'll still grow there anyway. And so, other bacteria have different ways of doing things, but this is just kind of a flavor of how bacteria are avoiding being killed by phagocytes. So I've talked a lot about bacteria, and again, I kind of mentioned that they're kind of small, and obviously they offer their own challenges, but what sort of large particles do macrophages or phagocytes need to eat? And one of them is referred to as apoptotic bodies. So in general, cells die of 2 ways. Necrosis, which is more of a spilling mechanism, cells lyse. It's very inflammatory and can cause problems due to this inflammation. The other way is often referred to as programmed cell death or apoptosis. And so every day, our bodies, about 10 billion cells die from apoptosis, and it's just part of the normal housekeeping of our bodies. They're regenerated by neighboring cells or precursors, but all of this material needs to be dealt with and cleaned up, and it's done by white blood cells or macrophages. And so here is, as opposed to just spilling its contents like a necrotic cell, it actually breaks up into small apoptotic bodies. And these are bigger than bacteria, and so they represent, a pretty good sized molecule that the white blood cell has to engulf. The immune system also has to deal with other invaders besides bacteria, and some of those are fungal pathogens or fungal spores. And so here is an image again, of a white blood cell that's in the process of phagocytosing these spores from Aspergillus, a common sort of household mold. So far we've seen that the macrophage, everything we've thrown at them, they've been able to internalize them pretty good. They can degrade most, though some give them problems. But a sort of interesting idea is, well, what happens if the macrophage actually runs into a particle that's too big for it to phagocytose? And so it's a process that we refer to as frustrated phagocytosis. And I show a picture of an egg here, but if you kind of think about the cell, if the yolk is the nucleus, and if you drop an egg in a frying pan, it really spreads out and takes up a lot of area and becomes very thin. And that's actually what, if presented with the right prey or pathogen or substrate, that's what happens to the macrophage as well. So here, we coat cover slips, so just pieces of glass with antibody that we usually use with the microscope, and then we parachute cells down onto them and then watch them spread out. And so, again, we're using a GFP-tagged actin here. And so we're actually observing them now from below. And so what you'll see is that there's an initial point of contact, much like the egg in the frying pan. It will hit, and then it will spread out until it kind of reaches a point where it can't spread out anymore. And so as this is going on, actin ends up polymerizing around the edge, and the cell continues to spread. Until finally it basically reaches its limit. And so this process is pretty complex. It's also accompanied, as actin is polymerized out here on the edge, and at these contacts, it's actually cleared and turned over from the middle. But this seems to be a fairly good model of frustrated phagocytosis, and there are some instances where we see these sorts of things again in the human body, and a very important type of frustrated phagocytosis would be involved in osteoclast remodeling bone. And so, again, bone constantly is undergoing synthesis or production by osteoblasts. Osteoclasts are the other part of the equation. They're basically fuse together macrophages, so they make a giant cell, and their basically job is to, pancake up against the bone and form a tight seal, and they're able to slowly degrade the bone, and they kind of move along, and just part of the normal turnover of the bone mass. Other types of frustrated phagocytosis deal with when neutrophils and macrophage encounter different types of hyphae. And so before I showed you a picture of a cell eating Aspergillus spores, but when the spores start growing again, they end up forming these long hyphae. And you can see here that there's a phagocyte basically wrapped around the length of the hyphae because it can't really internalize the whole thing. So it's sort of spread out and up and down this structure. Another fungal pathogen that forms hyphae and actually causes is the cause of yeast infections and thrush in infants is referred to as Candida albicans, and it can also form long hyphae. So here's the body of the Candida here, and it's formed this long hyphae, which this macrophage here is now trying to engulf, but it really can't eat the whole thing. And if it goes long enough, I mean, this hyphae can kind of poke out the other side, and so this is sort of another situation where, despite being great eaters, these cells do encounter structures that they have trouble dealing with. And so so other examples of this would be bacterial biofilms, or even things like asbestos, though it's not so common anymore. Asbestos used to be a real problem. It was used as insulation, but breathing it in, it would cause cancer and things like that. And it's very fibrous as well, and much like with the fibers here of the fibrous sort of shape of the aspergillus, the macrophage and neutrophils would end up trying to engulf these large filaments of asbestos, but couldn't do it. So maybe some of you guys are wondering, what happens? Can macrophage actually eat so much that they can't digest, or do they ever eat something that they don't have the enzymes to break down? And so there's a few examples of this, but not a lot that I could think of offhand. But there's this one study from about 10 years ago now. And I talked before about proteases and lipases being important in the phagosome for breaking down bacteria and pathogens. But in the case of apoptotic bodies, or in this case, red blood cell nuclei, the macrophage actually need this DNA 2 enzyme to degrade the DNA. So red blood cells are some of the unique cells within our body in that they don't have any nucleus. They start off with a nucleus, but as they're leaving the bone marrow, the nuclei are extruded, and then as they're in the bloodstream, they're basically just the leftovers. And so as a result, the macrophage in the bone marrow have to go around and clean up all this excess DNA. And so that's what we've got a picture of here. It's an electron micrograph. This is the nuclei of the macrophage, and you can see all these sort of expelled red blood cell nuclei here within the cell, and it can't degrade them because it doesn't have this enzyme. And it was kind of interesting, the identification of this mouse led to a lot of other studies, and this inability to clear the equivalent of apoptotic bodies, I guess, are very similar. These mice ended up developing things like arthritis and lupus and even cataracts in the eye. So it does sort of highlight that the importance of macrophage just going around and cleaning up debris in the body. All right, so we're almost there. But so another question that people have asked, or maybe it seems like sort of an interesting question to ask, is that do macrophage actually regurgitate after a meal? Do they ever eat something that they have such a dislike for they spit it back out? And so, yet again, this is another fungal pathogen, and it causes a meningitis. It's referred to as Cryptococcus neoformans. And these guys are super tough. What you can see here is that within the center here is the actual cell, and it's surrounded by both a capsule and a cell wall, and it contains melanin, and so they're very tough. They really resist killing by macrophage. And so, okay, it's going to project better than I thought it would. So this is a movie that was published a few years ago from a different lab, but what you're going to see is that their macrophage is going to take up some Cryptococcus, and after a while it's going to spit them back out. And so the macrophage eats. And so you can see the phagosomes in there now. The Cryptococcus is still alive. It's actually growing. And then finally, they're all expelled out. And yet the cell is fine because it's going to start, it actually divides and it goes around, it starts trying to eat them again. So it's not as if the cell's just rupturing and letting all these fungal pathogens escape. It's actually an active sort of spitting out process. And the really interesting, or something that I find very interesting, is that these cells, they can feed them both latex beads as well as the Cryptococcus, and they're selectively going to spit out the Cryptococcus and retain the latex beads. So here The latex beads are in green. You can see the Cryptococcus here as in white. And if I just play this, again, you can see the cell has both the fungus and the latex beads inside. And after a while, it just spits out the yeast, but keeps the latex beads. So people have referred to this as Vomocytosis, which, I don't know, is kind of a cute name. I don't know if I care for it, though. But that's sort of basically as many analogies to eating as I can make, I think, in 45 minutes. So just to kind of give you guys a quick recap, phagocytes, especially macrophage, are very good at eating various pathogens, invading bacteria, fungal pathogens, spores, but also other unwanted, we'll call it junk, but apoptotic bodies, the nuclei from red blood cells. They will try and eat different asbestos and uric acid crystals, and whatever you throw at it, they'll almost try and eat. They'll almost try and eat it. They're a really important bridge between the innate immune response and the adaptive immune response through the presentation of antigen to other cells. And you can really think of the lysosomes or the phagolysosome in these cells as the cell's stomach. It's acidic. It's where all the enzymes are to grade the particles they're taking up. But it's also important to know that successful pathogens, so the ones that are able to cause disease, are able to circumvent this killing through a number of means. So I'm going to finish off there. I was going to say, after all that talk of eating, is it lunchtime? But everyone's already eaten, I think, except myself. So I'll leave you with this here, and it's two other Yogi Berra quotes. I'll let you guys read them there, but I'd be happy to take any questions you have, and I actually will give a try at answering them. So.
Thanks very much. I just wanted to introduce myself. My name is Ori Rothstein. I'm actually Surgeon-in-Chief here at St. Michael's Hospital, but the other role I play here is the Director of the Keenan Research Center, which is where we do our basic science research. And first of all, I want to thank Greg for a really fascinating talk. We recruited Greg here, I guess it was about last summer, right? Yeah. Because Greg does really very fundamental biology. And I think what you saw here today was some really fundamental biology to help us understand how the cells that circulate in our body actually can engulf bacteria and other stuff to protect us from infection. And that's one of the key components of the Lee Cushing Knowledge Institute, because what we want to do is have people like Greg working with people like me who do clinical work and scientific work, and clinicians who work in the hospital all together, so we can kind of translate some of the work that Greg does into the clinic, and hopefully we'll be able to help patients. So I'm going to ask the first question. I know you had a question. I'm going to ask the first question. And my question is that, you've done a lot of work on how bacteria and other microbes are engulfed. Can you give us some insights into how kind of the findings that you have been translated, at least conceptually, not necessarily into the clinic, but towards the clinic? Because that's really what we want to do here.
Right. So I think some of the work that we've done, and I haven't talked about so much today, I think goes back to the way that certain types of bacteria they almost steal the phagocytosis process. So they'll come up against a normal cell that has no ability to kill them, and they'll inject infectors into the cells, and they'll basically induce their uptake. So they're causing normally non-phagocytic cells that can't degrade them, to take them up, and that's through changing the lipids in the plasma membrane. I think one of the other things that we've realized more recently is that kind of giving you guys a feel today that not all phagosomes are the same, and something that we've found recently is that not all phagosomes produce the same amounts of superoxide and the reactive oxygen species, and we've actually been able to ramp up and down their production through altering some of the lipids and some of the membrane components. And it may help explain why a small percentage of bacteria aren't killed by macrophage, because perhaps they end up in a phagosome that's not producing superoxide, things like that. So I guess those would be some of the to really help understand some of the pathology of invading bacteria.
Yeah, Greg went through all those different bacteria that are able to actually prevent themselves from being killed in the normal cells. Tuberculosis is one of the biggest killers in the world, and they do it because they have an ability to kind of thwart your own cells from killing them. And I think the work that Greg and colleagues of his do on understanding how tuberculosis works really have potential implications on altering our own cells and improving their ability to kill those bacteria, which, it's not a big deal here in Toronto, but around the world, tuberculosis is a real killer and very important. So why don't we open the floor to questions, and here's one right here. You have to announce yourself first.
Hi. I was wondering, this is kind of more of a medical question, I guess, but like all the symptoms of diseases that you see, like whatever, skin abrasions or anything like that, is that caused by the infector, or is that caused by the body's reaction to try and get rid of them?
Most of the time it's the body reacting and the recruitment of more different types of white blood cells, so a lot of cytokine production, things like that. So a lot of that is the inflammatory response that the body's sort of generating. It's the sort of, I guess from an evolutionary standpoint, it's short-term pain for long-term survival, I guess is the way this sort of system would evolve.
So just so you know, that's probably one of the most important questions in inflammation biology right now, because Greg talked the whole time about how these cells, the neutrophils, actually engulf bacteria and basically kill the bacteria as kind of their major function. But we know that those same cells, the leukocytes or neutrophils and macrophages, actually can release those molecules and injure your own tissues. So on his side, where you want to kill bacteria, what you want to do is juice up those neutrophils and macrophages and make them kill. So where I work over in the intensive care unit, we have 18 people there who are being ventilated and have all sorts of different diseases, and we're trying to cut back their inflammatory response, because it's actually, the inflammatory response is actually killing them and not the infection at all. So really kind of interesting biology, where good on one side, bad on the other. So your question is very relevant. When you get a scrape on your arm, there are no bacteria there, but you get inflammation. When you get a sunburn, when you think of it, when you get a sunburn, that's those neutrophils that are causing the sunburn, the ones that for a living kill bacteria, but when you get excessive damage from the sun, all of a sudden, they're harming you. So good question, thank you.
Yeah.
You mentioned a lot about how bacteria escapes the phagocytes, and I know it's a bit going off topic, but so is there any type of backup mechanism that the human body naturally has to kind of catch these escaped bacteria that can escape the phagocytosis?
Yeah, there's a lot of, I guess, I'm not sure if you'd call them backup, but there's sort of multi-pronged approach that the body has developed. I mean, one of the real frontline defenders of us from infection is skin and mucosal surfaces, which neutralize a lot of things right off the bat. But then there are antibodies in circulation that can immobilize bacteria, as well as complement, which is another way, proteins that recognize bacteria and can lyse them. I guess the other case, things that macrophage aren't necessarily good at killing, neutrophils tend to, because they're much more violent. Neutrophils are able to kill things that macrophage can't, especially some of the fungal pathogens. You really need good neutrophil function to kill some of those things. So there's I guess different weapons in the chest, but certainly there are enough pathogens that cause real problems. So, yeah.
When the lysosomes were aiding in decreasing surface area of membranes, how exactly were they doing that? Are they coming and melding with the membrane
so
and just sort of basically sacrificing themselves?
Yeah, there's very local fusion of the endosomes and lysosomes right at the spot where they're being internalized. So there's some other times that happens. It's sort of referred to as like a plasma membrane repair. If a cell is exposed to certain types of toxins, they'll start becoming leaky, and then lysosomes will go and fuse and patch up that area. So those organelles do have that sort of capacity to be trafficked and fused at those sites. Yeah.
My name's Helen, I'm a volunteer.
Okay.
When you're talking about TB all over the rest of the world, and it's what happens if the cell can't destroy the TB pathogen or whatever you call it? What? And you're TB positive, somebody's TB positive. What's stopping their flare-up?
Well, I think in the case of tuberculosis, especially, I guess, in the lung and the brain, they end up forming granulomas, which is much more than a single cell. It really becomes that you could have a little cluster of bacteria growing, and then, again, it's the macrophage and stuff kind of almost surround them, but it's a much bigger structure. It's almost like the cells are cutting them off from access to the rest of the body by building up a layer around them. But I'm not sure. Besides that, that's besides that, that's the really only thing I can think of. Yeah. Yeah.
Just on the last clip that you showed us,
Yeah.
where the feed,
Yeah.
also the cryptococcus,
Yeah.
but it's spit out cryptococcus only. So do we know why it only spits out cryptococcus?
No, that's actually something that I've been interested in, but if you kind of watch the time lapse of the movie, it's several hours afterwards, and so it becomes a challenge to study experimentally, and so that's been the biggest obstacle, I think, to really characterizing this. The observation itself is only about 7 or 8 years old, and so there's been some thoughts that maybe there's been some observations of what might prevent it from being spit out right away, but there really hasn't been anything worked out of what's controlling that mechanism. I mean, one sort of idea or theory is that it could be the similar machinery. So during the initial stage of phagocytosis, endosomes and lysosomes are delivered to the site to help with the eating. Now you have those phagosomes that are really akin to the lysosomes. Maybe there's some sort of signal that has confused the cell machinery, and so now maybe it thinks it needs to fuse that with the plasma membrane, even though it doesn't necessarily. But if the same machinery is used in both cases, and so as a result, once it fuses, the cryptococcus ends up back in the media. So, but really, there's really just scratched the surface, I think, on that whole observation. There's only a handful of studies that have even looked at it. So it's really unclear at this point. Yeah?
Just going back to the same video he just mentioned,
Yeah.
in that clip, you see that I believe it's a latex bead, right?
Mm-hmm.
You can still see it because it's lit up by GFP, right?
Right.
How come after a few hours it's still there, not degraded by the enzymes?
Yeah, so the latex beads, again, are one of those indigestible materials.
Okay.
They'll actually stay in there for a long time. We've actually taken advantage of that at times where we can sort of feed the cell latex beads, do something to the cells the next day, and then kind of look at what happens at the very late stages of phagosome maturation, things like that. So the latex beads hang around just because the cells don't have the enzymes to degrade them. So.
Yeah, I think latex, it's a pretty good tool for a model of phagocytosis because you can see them very easily under the microscope because they're so big, but also they don't get digested. And I think that those two things make it very easy to follow under the microscope. So people have been using those,
and they're safe. They don't make me sick.
Yeah, but I think you know one of the things that that Greg talked about was he talked about coating them. You have to actually prepare the latex beads. You have to kind of butter them, otherwise the neutrophils won't eat them.
Yeah.
So you have to prepare them for that. So it's a really effective tool for kind of studying the process of phagocytosis. So a lot of people use that.
I'll just, coming back to just one other thing, something that I didn't mention, is that the sort of regurgitation of those cryptococcus seems to be both part macrophage and then part cryptococcus, because if you can kill the cryptococcus and then feed them to the macrophages, and that regurgitation goes way down. It doesn't happen nearly as often. So it seems as if maybe those organisms are secreting something or doing something to signal from inside the phagosome to help spit them out. So it's even more complicated than just the cells themselves. So.
Okay, so it's our tradition to actually welcome groups individually. I know there are a bunch that are here. So I know there are University of Toronto students, I think. Who's from you? Anybody here? So first of all, one thing, the way you identify a U of T student is they'll never put up their hand when you ask them. So if you're from U of T, put your hand up. Yes, there you go. Okay. So we have people from Ryerson University. Okay, good. Thanks. Welcome. We have some of our volunteers who come on a regular basis, so thank you for coming. We have students from Branksome, so identifying for all here, and students from Upper Canada College. There, good, thanks. We have two students, I think, from Crescent School. They're here, they're in the back there. And we have a whole bunch of people who are kind of co-op students and volunteers at the hospital here, so put up your hand. Okay, so who did I miss? So first of all, thank you again for coming this afternoon. We really enjoyed you coming to St. Michael's and seeing our beautiful facility here. And thank you, Greg, for a really interesting lecture. The students who want to stay have two more components to the afternoon. So first,
that's separate discussed with specific students.
I see. Okay.
Yeah, that may time. So.
Apparently.
Yeah.
There are some other opportunities for people to go to the simulation center, and also we have a zebrafish facility, and zebrafish are kind of one of the interesting things we have here at St. Michael's. So they don't look anything like zebras, first of all. They're transparent, and they have no stripes. But we have a scientist here who uses the zebrafish with a view to actually studying various aspects of biology. The beauty of a zebrafish is that there are lots of them in an aquarium. You can breed them very quickly, but you can actually label specific proteins with specific fluorescent dyes. So if you want to look at the heart, you can put a dye in there to look at the heart and see how your interventions can actually alter the heart function. So you're going to see some of the zebrafish stuff. So what's my protocol from here on in?
We're good. No, we're good to go for now. So everybody knows about the tours that are important.
Okay. So again, thanks for coming, and